Welcome to The Explainer. Today, we're going to dive right in to one of the most fascinating and absolutely critical bottlenecks in the entire world of drug development, Phase 2 clinical trials. You can really think of this as the ultimate funnel of discovery. It's that exact moment where scientific theory finally collides with real-world human biology. Here's our quick agenda for how we'll see a drug prove its worth today. First, a welcome to Phase 2, followed by its core objectives. Then, we're going to look at the essential prerequisites, and finally, we'll wrap up by comparing Phase 2A and 2B. All right, Section 1, welcome to Phase 2, first in patients. So, the absolutely crucial thing to understand here is that Phase 2 is the bridge. It's explicitly categorized as a therapeutic exploratory phase. Basically, this marks the very first time a new drug is tested in actual patients, people who are actively suffering from the disease the drug is designed to treat. Up until this point, back in Phase 1, scientists were really just testing the drug on healthy volunteers, just to make sure it wasn't outright toxic. But here, the drug finally meets the patients who actually need it. The ultimate goal is confirming its effectiveness while strictly, and I mean strictly, monitoring for side effects. And that brings us to the scale of it all, shifting from just a handful of healthy volunteers in Phase 1 to a much more focused, statistically relevant group of about 20 to 300 actual subjects. This number isn't random. It's carefully chosen. It's large enough that researchers can start seeing real, measurable trends in how the disease responds to the drug, but it's still small enough that they can tightly control the environment and closely monitor every single patient for any unexpected reactions. It's kind of the perfect sweet spot for early testing. Now, as you might guess, this kind of rigorous testing definitely doesn't happen overnight. You just can't rush the scientific method. The duration of a Phase 2 trial can take anywhere from a baseline of 6 months, stretching all the way out to several years before the phase is actually completed. Researchers need this prolonged window to ensure absolute safety and effectiveness. They have to see not just if the drug works on day 1, but if it continues to work on day 100, and of course, if any long-term side effects start creeping in. Moving on to Section 2, core objectives, focusing on efficacy and safety. So, what are researchers actually looking for? Well, they divide their focus into 2 main buckets. The primary mission is quite straightforward, proving efficacy in patients. Put simply, does the drug actually cure or treat the disease it was built for? But the secondary, and frankly equally vital objective, is keeping a strict eye on patient safety issues. Remember, they are now dealing with sick patients whose biological systems are already compromised by disease, so the drug might act completely differently than it did in those healthy volunteers. But it's not just a simple yes or no does it work. Scientists also use this phase for what we call optimum dose finding. This is where things get highly mathematical. They're meticulously dialing in several variables. They study the dose-efficacy relationship to figure out exactly how much of the drug is needed to trigger a response. They have to map out the therapeutic dose regimen, the exact duration of the therapy, and the frequency of administration, like should you take this once a day, 3 times a week. Ultimately, the whole shebang is about identifying the perfect therapeutic window. That's that exact, precise dose range where the medication is strong enough to do its job, but low enough to avoid toxic side effects. Okay, Section 3, prerequisites and mandatory approvals. Now, you might be wondering, what actually happens before Phase 2 even begins? How do we even get here? It's a great question, because the prerequisites for starting this phase are absolute and entirely non-negotiable. The medical community takes human testing incredibly seriously. Before a single sick patient is treated, innovators and experts must rigorously review all that safety data generated from Phase 1. They have to absolutely prove the drug didn't cause undue harm to those healthy volunteers. Then, securing prior approvals from regulatory ethics bodies is strictly mandatory. You literally cannot move forward without a green light from the Institutional Review Board, or IRB. And, depending on the jurisdiction, like the specific framework from our source, prior approval from national authorities such as the Drug Controller General of India, or DCGI, is also an absolute requirement. Basically, no approvals, no trial. Simple as that. Actually, scratch that. There is one major, fascinating exception to this rule. What if researchers want to test a completely new use for a drug that is already out there on the market? Well, because that drug has already been extensively tested and proven safe for human consumption in its original trials, researchers actually get a Phase 1 exemption. They're allowed to skip testing on healthy volunteers and fast-track directly into Phase 2 to see if the drug works on this new disease. Talk about a real game changer for speed. And finally, Section 4, comparing Phase 2A and Phase 2B. So as phase 2 progresses, it actually splits into two distinct flavors. Phase 2A is the early phase. It functions as a smaller pilot clinical trial, strictly designed to assess those dosing requirements we talked about. It usually involves just 20 to 200 patients. It's often a single-blind comparison against a standard drug, meaning the patient doesn't know what they're getting, but the doctor does. And it's generally not multicentric. It might just be run at a single research hospital. But once they lock in that dosage, they graduate to phase 2B. This is the late phase, stepping up into a much larger pivot clinical trial that is wholly designed to study efficacy. The patient count scales way up, hitting between 50 and 500. The scientific rigor also tightens up significantly. It becomes a double-blind trial compared with a placebo or standard drug. That means neither the patient nor the doctor knows who is receiving the experimental treatment, which totally eliminates bias. Finally, its scope becomes multicentric, spreading across various geographic locations to ensure the data is diverse and incredibly robust. When you put it all together, phase 2 truly is the ultimate crucible of medical science. It's the strict, highly regulated bridge between basic laboratory theory and widespread patient cures. It's where we finally find out if a promising idea actually has the power to heal us. I hope this explainer helped you understand the intense rigor our medicines go through to keep us safe and effectively treat disease. Please like, share, and subscribe our channel for more deep dives. And it leaves you wondering, with regulations this tight, what other medical breakthroughs are waiting just on the other side of the funnel?